Well, first of all, thanks, Neil, for inviting me to talk and share a little bit about some of the activities going on at the University of California at Berkeley. Oops, sorry, can I just spin it this way, because I stay in front of the podium. So this is a relatively new initiative at Berkeley, and the goal was to bring together the widest swath of researchers on campus who have been looking at a variety at different scales, a variety of different ways of looking at how species have changed over time, and then more broadly how systems on Earth have been changing over time. So when I say broad, it's encompassing up to 8 different departments on campus, including biological sciences, which, organismal and molecular biology, earth sciences, climatologists, paleontologists, and a variety of also our natural history museum collections, as well as field stations. So it's a rather ambitious project to bring us all together. And what this initiative has looked like so far has been essentially a variety of symposia, where we've been able to come together and produce, oops, sorry, I forgot this is the button, a couple of review papers that have been really helpful in advancing what we know so far in terms of sort of the global change biology picture. This one was produced by about 22 researchers across campus, and was looking at specifically state shifts. And like I said, it made kind of an impact in the way we started looking at not just what we're doing on campus, but also how we're thinking about global change research. So I will talk to you about one of the many funded projects so far. It's actually one of 2 funded projects, but one of many specific research projects that have been initiated. This one was funded by the William Keck Foundation, and we're calling it the Keck Bioinformatics Engine, or the Keck Engine. Our co-leads, our main PIs are Rosie Gillespie and Charles Marshall, and then we have a variety of also co-PIs, and you can see just from our affiliations that we are sort of a cross-section of organismal and Natural History Museum collections, and this kind of is indicative of sort of the broad collaborative nature of all the Big CB projects. So this one in particular, I'll spend a little bit more time talking about. Our primary aim here is to mobilize some of the rich sort of natural history repository information that we have in Berkeley. They may be in Natural History Museum collections. They may be locked up in our field stations in the forms of archival field notes, in the forms of maps, experimental results, and climate station and high-resolution networks that are being started on a variety of California field stations, but it also includes data sets that have been in maybe a principal investigator's lab on his local hard drive, for instance. And so we have a variety of these. These also includes things that have been stored away in university facilities that have yet to be fully cataloged and vetted and digitized for access. We're also going to marry this with GIS data stacks. This includes both paleo, present, and future climate model scenarios, remote sense data, and hydrology models, just to give an example. And the main goal then will be to bring all this together and visualize it in a web-based portal. And we believe that we won't be able to fully understand the drivers of change over time unless we're able to integrate and visualize all these different assets and understand how each are affecting each other and those responses. And so this could be at a lot of different levels within food webs, larger ecosystems, perspectives. It could also be at much more specific levels of molecular and genetic impacts and how that is expressed phenotypically, and then thus into larger impacts of community and species compositions changing and shifting through time. So let me just move on here, and I'll briefly go through some of our assets here. So, you know, like I mentioned the 4 museums here that we have. Together, we've got about 15 million specimens for each individual collection. We've got 10 to 100% digitized and georeferenced. So what this means, though, we do have some major challenges ahead of us. And like I said, our first main aim is to actually get a lot of this information digitized and databased and accessed in the GIS. This also includes a lot of non-traditional specimen, but the sort of thing that we've been talking about, contextualizing data. So more specifically, this might include things like historic images that have been georeferenced. These gives us opportunities to do photo retakes. We have also pollen core samples, and then moving from sort of specific landscape level, we can also look at regional maps that were hand-drawn in the 30s and have now been digitized and put into a geodatabase. And this, again, could be married with high-resolution LiDAR or other remote sense data. So what the infrastructure kind of looks like on the informatics end is basically being able to allow the collection to continue to do their primary collection management in their individual databases. We're going to integrate these actually in a cloud-based platform. And in this proposal, we're really very interested, and this is sort of an ongoing theme, of leveraging all sorts of other currently in progress informatics projects like VertNet, another NSF project that will be allowing us to access all our data sets in through web services. The actual platform itself is fairly straightforward, where we grab the collecting events. So everything is kind of place-based research. So here's our place-based specimens. Those are going to be intersected against GIS data stacks, and this will allow visualization within a common portal of all these disparate data sets that usually takes at least 3 or 4 different principal investigators to actually access and get them in one place where we can easily do charting, share them, but more importantly, start trying to identify these drivers of global change. And I think I've run out of time, so I will talk to anyone more specifically later.